Good evening, everyone. My name is Andy Rachleff, and I am the chairman of Damon Runyon's board of directors. On behalf of our entire team, I welcome you to our 80th anniversary celebration. As you are about to learn, Damon Runyon is very different from any cancer research funding organization with which you might be familiar. We only fund young scientists because the data is very clear that all of the breakthroughs in the arts, humanities, and sciences come from young people. We don't focus on specific cancers like everyone else because breakthroughs come from following the surprises that come from experiments, and those surprises almost always lead our investigators to different cancers. So as a result of our unique approach, our scientists have been involved with more than 80% of the breakthroughs in the last 30 years in cancer research. What's even more amazing is our funding is less than 0.3% of annual US cancer research funding. So think about that as a power law. We provide less than 0.3% of the total US cancer research funding, yet our scientists have been involved with more than 80% of the breakthroughs. No other cancer research funding organization can make anything close to a claim like this. And it's for this reason that we honor scientists at our annual event, not philanthropists. Another thing that makes us very different. Now, this evening, you're going to hear from a number of scientists who will explain how pursuing high-risk, high-reward research leads to these great breakthroughs. Now, I believe the Damon Runyon approach is superior to anything anyone else is doing, but don't take my word for it. Please ask one of our funded scientists who is seated at each and every one of our tables. And I think you will find a very consistent story. Now, with that, I'd like to invite our CEO, Young Lee, to come up here and tell you about the rest of our program. Thank you very much.
Good evening. I'm Young Lee, the president and CEO of the Damon Runyon Cancer Research Foundation. I'm truly honored to welcome you here this evening. Thank you so much for joining us on this celebration of our 80th anniversary and the incredible breakthroughs our scientists have made since our founding in 1946. The past 80 years of Damon Runyon-funded cancer research are a testament to the impact that focused and strategic funding can have to reshape our entire approach to understanding and treating cancer. Our scientists have made amazing advances and impact in all areas, from the earliest understanding of how genes and viruses are linked to cancer, to the very first uses of chemotherapy and radiation and bone marrow transplant to treat patients, to, in more recent years, development of novel immunotherapies and targeted therapies and new applications of artificial intelligence. Just this week, data were released about a new targeted therapy that can extend survival for pancreatic cancer patients. This is truly extraordinary. We're seeing the success of novel vaccines that can actually prevent certain cancers, and Damon Runyon scientists have made critical contributions to each and every one of these areas. Today, the Foundation funds research through 8 different award programs, each one targeted to support a specific cohort of scientists, from basic researchers to quantitative biologists to pediatric cancer researchers to physician scientists. We currently fund nearly 200 scientists across the country, and each one is working to tackle the most difficult questions and challenges to better understand how to personalize cancer treatments, diagnose cancers earlier, and prevent them altogether. The impact of cancer research is tangible and huge. Thanks in large part to our scientists' incredible and invaluable contributions, the cancer survival rate has improved by 33% in the past 30 years. This has led to an estimated over 18 million survivors in the United States alone. But we have much more work to do. The odds are still that 1 in 2 men and 1 in 3 women will receive a cancer diagnosis in their lifetime. These are scary statistics. We need to continue to invest in research so that we can ultimately eradicate suffering from all cancers. As some of you know, cancer came close to home for me 4.5 years ago when my husband was diagnosed with an advanced aggressive sarcoma, a rare cancer of the bone and connective tissue. It was truly devastating, and he passed away 3 years ago. I know many of you in the room have similar stories, whether as patients or caregivers or supporters of loved ones. Many of us have unfortunately experienced the fear and uncertainty that surrounds a cancer diagnosis. Cancer is an incredibly complex and unforgiving disease that does not discriminate. Yet, I will always appreciate how fortunate we were to have the wisdom, care, and support of our oncology community here in New York City and across the country, including some of the scientists and doctors here in this room. They gave us hope and trust in science and medicine. As a result of my experience, I view myself as an advocate for patients, but long before that, also as a scientist and as a researcher. I was myself a Damon Runyon Fellow, and so I very much understand firsthand the challenges that a young scientist faces. This is a very demanding career path, and I personally remember the invaluable support that I received from Damon Runyon, my peers, and my mentors as part of this distinguished scientific community. Damon Runyon's mission, funding brave and bold, is more critical than ever. We want to maintain optimism and hope for the future, but many challenges lie ahead. We're living in unprecedented times when progress in the future of science and medicine are threatened by funding cuts and policy changes. The path for young scientists in the United States has never been rockier than it is right now. And we risk losing the next generation of leaders, not just in cancer research, but in all areas of science, as they make the decision to potentially pursue careers in other more stable fields or leave the country altogether. For decades, we have led the world in making life-saving discoveries, but now our position as the world's leader in science is under threat. But even in this difficult moment for science and in this country, there is much to celebrate and look forward to. Thanks to all of you and your very generous support, Damon Runyon will continue to enable the next generation of leaders in cancer research to take risks and pursue innovative paradigm-shifting studies that have the potential to improve treatment, diagnosis, and prevention of all cancers. We equip them to follow the science, embrace serendipity, and savor the surprises. Our mission has never been more important than it is now. There's no better representation of our ethos and mission than tonight's honoree, Dr. Matthew Vander Heiden. As a Damon Runyon Fellow and later a Damon Runyon-Rachleff Innovator, Matt sought to answer the long-standing question of how cancer cells fuel themselves. Today, cancer cell metabolism is an incredibly active field of research, and he is considered one of its pioneers. Proof that the Damon Runyon model is exceptional in identifying tomorrow's leaders. In addition to his scientific accomplishments, he's a selfless mentor and has gone on to sponsor multiple Damon Runyon scientists in his own lab, as well as volunteering his time to serve on our award selection committees. We at the Foundation are incredibly proud of his extraordinary accomplishments and are honored and grateful to consider him a friend. Our 80th anniversary gives us cause to reflect not only on the incredible scientific legacy, but of our visionary leadership as well. This year, we are especially delighted to announce the launch of the Walter Winchell Society to honor those emeritus trustees who have shaped us over these past 80 years. Our foundation began in 1946 with the famous radio personality Walter Winchell, who went on the radio after his good friend Damon Runyon died of throat cancer. He put out an urgent call to Mr. and Mrs. America to give their spare change to support cancer research. He and the original trustees had the vision to support young scientists, and over the decades, the board chose to prioritize the possibility of breakthroughs over incremental progress, and our legacy of impact is a direct result of their bold courage. Through the Walter Winchell Society, we honor those who have made the Damon Runyon Cancer Research Foundation such an exceptional success. We congratulate and honor those in the room with us tonight. And David Marshall, Sam, I'm not sure where you are. Sam Silverstein. And Craig Thompson. We congratulate all of the other members of the Walter Winchell Society as well, and they are listed in your program. We're so grateful to all of them. Our current board of directors follows in their footsteps. We're truly grateful to Andy Rachleff for his leadership of the board and to each one of our board members. Their support is absolutely critical to the longstanding success of our organization. I'd like to ask all of our board members to stand up or raise your hands, please, so that we can recognize you. Thank you all. So I want to especially thank the Damon Runyon staff for their dedication and hard work each and every day. And so on behalf of the entire staff and board of directors, I want to extend a very special thanks to Claire Cahill, who Claire is our Chief Administrative Officer, and I think each and every one of you, and especially all the scientists in the room, have had interactions with Claire over the years. And so she has spent over 40 years leading our award programs and overseeing countless other responsibilities at Damon Runyon. It's difficult to imagine a version of Damon Runyon without Claire, and we have been incredibly fortunate to benefit from her wisdom, her clarity, and perhaps most of all, her good sense of humor for all of these years. We're truly grateful. Thank you, Claire. And so for the last of my thank yous, at the moment, we're inspired by our Damon Runyon scientists, present and past. The work that they do each and every day represents promise for future breakthroughs, and we're proud and honored to support their research. We're grateful to our scientific committee members who generously volunteer their time and expertise to select the next generation of leaders in cancer research. So at this time, I'd like to ask all of our current and former Damon Runyon scientists and committee members in the room to please stand so we may thank you. So we are so grateful to all of you. I think many of us talk about how Damon Runyon is like a family, and hopefully you all feel that in this room. We are, I think, very much connected to each and every one of our scientists, to our supporters, to our board members, and so grateful that we are able to work together in this with our very important mission. So it is now my honor and privilege to introduce Dr. William Kaelin. Dr. Kaelin is the Sidney Farber Professor of Medicine at the Dana-Farber Cancer Institute and Harvard Medical School, and is a Howard Hughes Medical Institute investigator. He has many, many scientific accolades, but most notably, he was awarded the 2019 Nobel Prize in Physiology or Medicine for unraveling a molecular mechanism that explains how cells sense and adapt to varying levels of oxygen. These findings are leading to new therapeutics for a wide range of disorders, including cancer, cardiovascular disease, and anemia. We are so fortunate to have had Bill's involvement in the leadership of Damon Runyon for nearly 20 years. He initially served as a member and then chair of the Damon Runyon Clinical Investigator Award Committee. Bill joined Damon Runyon's board of director and was elected vice chair of scientific programs in 2016 and currently serves as the chair of the Physician Scientist Training Award Committee. We are so privileged to have his support as one of Damon Runyon's greatest advocates and a true champion for young scientists. I'm delighted to turn the podium over to Dr. Kalin to share his thoughts.
Well, thank you, Young, and thank all of you for being here. So I grew up on the South Shore of Long Island. Go Knicks, by the way. And so I spent a lot of time with my father fishing. Now, my father, as it turns out, was first generation college, and his father had been a manual laborer who had laid reinforcing steel for roads and bridges around New York. I used to tell people my grandfather built the Verrazano Bridge, which technically was a true statement, but maybe a little bit of exaggeration. But there are a lot of similarities between fishing and doing science, starting with the need for patience and a lot of luck. But the other similarity is you can only learn so much about fishing by reading a book. In the end, you need to get out on the water, and preferably with a wise, seasoned fisherman who really knows what they're doing, who can teach you not only the techniques of fishing, but more importantly, can start to pass on to you their wisdom, their instincts, and their intuition. And the same considerations apply to science. So if I could have the first slide, I think I have one and only slide, but if we can see, get our first slide. Yeah, so I heard some appreciative awing because many of you know, yeah, how about his wife? We're gonna get to you in a minute, Emily, don't worry. So the person who mentored me and enabled me to become a scientist was the great David Livingstone, who many of us knew and loved, but I certainly owe my scientific career. Yes, let's. And you stole my thunder. I was going to say, and his beloved wife, Emily, is with us this evening, so a round of applause for Emily. But David was one of the greatest scientists of his generation. He was brilliant and kind and masterful, and he loved the Damon Runyon. In fact, I think he spent over 20 years either as a consultant or as a director for the Damon Runyon Cancer Research Foundation. And he frequently would describe the Damon Runyon Cancer Research Foundation as a national gem. And that's exactly what it is. And what he meant by that was, as you probably know, there are many cancer charities that focus on a particular cancer or on a particular institution. But as you just heard from Andy, that's not our formula. Our formula is we find the best and brightest young people wherever they happen to be, and then we make sure they have great mentorship. Again, mentorship's important, and we make sure they have the financial resources to succeed. The other thing we do, which is really important and part of our secret sauce, is rigorous scientific peer review. There's the saying, A's hire A's and B's hire C's, and that's very true even in scientific peer review. So I can tell you we're blessed to have scientific peer review committees that are populated by the leading scientists in the United States. And so this enables us to, again, find the best and brightest and place those bets. Now, I can tell you, if you are lucky enough to win the Nobel Prize, you suddenly magically become very popular. So I've learned I really have to protect my time. I'm getting better at saying no. I'm not quite there yet, but I'm learning how to say no. But I can't say no to this organization because I believe in this organization. I've seen what it can do. I know it punches above its weight consistently in moving the field forward, as you so nicely heard from Andy. And it's absolutely the truth. Now, this is very personal for me. Some of you know that my late wife, Carolyn, was a brilliant breast cancer surgeon who ironically developed breast cancer herself in 2003. Thankfully, she was a survivor of that cancer, but in 2010, she developed a malignant glioblastoma. We were told she had about 1 year to live. Thankfully, with the help of a lot of people, including some of the people in this room, we put together a number of experimental therapies, and we were able to keep her for 5 years. So it wasn't long enough, but it was something that at least I cherish to these days, that we had her for those 5 years. But I can tell you now, I'm absolutely determined that this doesn't happen again. I can't tell you how painful it was as a cancer doctor and as a cancer scientist to realize that with my beloved wife, I was now out of bullets and I was going to lose her. And I said to myself, we can't let this happen for our children and our grandchildren. So this is my mission for the rest of my life, is to make sure we keep pushing the field forward so that we never face these sorts of dilemmas with our loved ones in the future. Now, I can tell you in that regard, Carolyn was the quintessential mama bear who was always protecting her cubs. So I have no question that if she was still alive, she would be in Washington right now giving certain people an absolute earful. And I can tell you why. First of all American labs, including my lab, require a steady stream of talented foreign trainees, and that is down to a virtual trickle. We also require stable, predictable federal funding to complement philanthropic funding, but now that's become questionable. And finally, even as of this week, there's now a proposal to take scientific peer review in the United States for federal funding and put it under the control of non-scientific political appointees. So all this is to say, she'd be down there saying, would you get out of my husband's way because he's trying to do some good in the world and you're making it much more difficult for him. So I tell you this because there's never been greater importance to the Damon Runyon as far as I'm concerned, because as you just heard, this is a challenging moment, as Young has outlined, but we're here trying to keep fighting the good fight, pushing the field forward, and making sure we don't lose the next generation of these magnificent young people who have dedicated their lives to curing this awful disease. So thank you again for being here. I can't thank you enough, and it means a lot that you're here because, again, the scientists of the world are getting all sorts of messages, and when you're here, you're saying, we believe in you. Thank you.
Thank you so much, Bill, for your wonderful comments, as always. We are going to take a break from the program, and the entrees will be served. So please enjoy conversations with the scientists and other guests at your table, and then we will resume the program shortly. Thank you. I'm so happy to see that everyone is having lovely conversations. I'm sorry to ask you to take your seats so we can resume our program. So before we celebrate our distinguished honoree, we're delighted to have the opportunity to hear from several of our scientists who will share their work with you today. So it's my pleasure to introduce our first scientific speaker this evening, Dr. Kelly Bolton. Kelly is an assistant professor at Washington University School of Medicine and a Damon Runyon clinical investigator. As you'll hear, her work on cancer risk factors has opened new possibilities in clinical trials and is shaping how we think about cancer prevention. Please join me in welcoming Dr. Bolton to the podium.
Thank you so much. Yeah, my name's Kelly Bolton, and I'm so happy to be here and so thankful to Damon Runyon for all their support of me over the past few years in starting my lab. And a lot of the work that we do is really trying to identify people who are at a high risk of getting leukemia and trying to prevent leukemia instead of treating it when it's already very difficult to treat at that point. And so targeted therapies that are gene-directed have been really revolutionary in cancer care. They've been used across solid tumors and hematologic cancers now. And the promise of them is really that because they're targeted and they have less side effects, Maybe people would tolerate them better. We could give them for longer periods of time. The issue is that because cancer is so complex, we often can't give targeted therapies alone. We still have to give them in combination with chemotherapy. And the reason for this fundamentally is that cancer has these built-in mechanisms for resistance by the time you're diagnosed. So most cancers aren't driven by just one genetic alteration. It's usually at least 4 to 5. And because there's multiple mutations that are driving the cancer, targeting one mutation can unfortunately sometimes lead to the emergence of other mutations that then lead to resistance. One example of a targeted therapy that's been really fantastic in leukemia care are IDH1 and 2 inhibitors. The IDH1 inhibitor ivosidenib is a targeted therapy against a mutation in IDH1, which is a cancer driver in myeloid malignancies, blood cancers, and also solid tumors. These drugs really, they have basically no side effects. They're really fantastic drugs, very safe, and they have pretty good response rates, like 30 to 40% of people respond, but unfortunately, most people will relapse at some point while taking the therapy. And as I was alluding to before, the real driver of this is that you have the IDH1 mutation, you probably have a couple co-mutations along in that cell, and the mutation will decrease over time when you give the targeted therapy, but then you have emergence of a second or third mutation that will lead to relapse and to the patient no longer responding. So, but obviously you look at this picture and you might already think, well, what if we were able to identify people before they had overt leukemia and to treat them with these targeted therapies early when they had maybe 1 or 2 mutations driving the abnormal clone. And we're actually really lucky in that in the space of leukemia, we actually really understand the natural history quite well. So people, when they're middle-aged, often have these mutant blood cells that have leukemia driver genes. And over time, these mutations expand, they acquire multiple mutations in the same cell, and then eventually they overtake the bone marrow and you can start to get abnormal blood counts. So when you get abnormal blood counts at this phase, people are at super high risk of developing leukemia. So at least for people that have IDH1 mutations and abnormal blood counts, we think it's over 50%. So knowing this, we thought that maybe if we can target people in this phase where they have abnormal blood counts and IDH1 with the IDH1 inhibitor ivosidenib, we could actually prevent them from developing leukemia and lead to long-term cures. So with this in mind, we developed the PIMS trial, which was supported by the Damon Runyon, and where we gave ivosidenib to 20 individuals with IDH1 mutant CCUS, with its total cytopenias of uncertain significance, with the outcome, I said that really quickly. I should congratulate myself on that. And the outcome was a hematologic response. So did we resolve their abnormal blood counts? And the issue with this trial always was, when we were thinking about it and planning it, is that it's not always straightforward to find these individuals. So we do a lot of sequencing in people that have abnormal blood counts, but even then, it's an underrepresented entity. People aren't always aware of it. So we knew that finding these people would be an issue. So we actually launched this as a decentralized trial, which means that patients all over the US were eligible to participate. At WashU, where I'm an independent investigator, we were the study site, but we actually shipped the drug directly to patients in their homes. And all the study activities took place remotely. We did remote study visits and remote labs and EKG monitoring for safety. And the trial has worked really, really well. We've enrolled after a couple of years, and we had really amazing geographic and socioeconomic diversity because of this decentralized trial where people, it really lowers the barriers to participating in a trial. So we've had really remarkable responses. So 90% of the patients had a hematologic response, so they had improved normalization of their blood counts. And we've had only 2 out of the 20 over a few years of follow-up now have progressed to hematologic cancer. So we think this is a big win. And we see that this is really immediate. So right when you give the drug, you have this rapid normalization of blood counts, and it's sustained over many years now of treatment. And the other important thing is that over time, we are able to get rid of the IDH mutant clone, which suggests that we are able to reverse the natural history of these mutations and to prevent these clones from expanding and developing into leukemia. So in summary, with the wonderful support of the Damon Runyon Foundation, we've shown that this is a really promising idea, I think, using these targeted therapies in people to prevent cancer before it develops. It seems like it works better, perhaps, in this setting than when people have overt malignancy driven by multiple mutations. And our next step is we're actually trying to move this even earlier to identify people before they have abnormal blood counts to treat them where we think that we might even have higher success rates of preventing these mutations from expanding and progressing. So thank you.
Thank you so much, Kelly. For our next scientist speaker, I'm pleased to welcome Dr. Mark Yarmarkovich. Mark is a faculty member at NYU Perlmutter Cancer Center at the NYU Grossman School of Medicine and a Bakewell Foundation Damon Runyon-Rachleff Innovator. Mark leads a laboratory working on next-generation CAR-T immunotherapies, which he will explain to you, capable of precisely targeting tumors in a wide range of cancer types. Please join me in welcoming Dr. Yarmarkovich to the podium.
Thank you very much, Young. So my name is Mark Yarmarkovich, and I'm really honored to give you a little bit of a snippet of our work funded by Damon Runyon, our Rachleff Award, supported through the generosity of the Bakewell Foundation. So in 2012, a young girl named Emily Whitehead had exhausted all of her options for a leukemia. Her family was told that she had a few weeks left to live, and they were offered an experimental therapy called CAR T cells. Following a single dose of CAR T cells, Emily woke up cancer-free and remains healthy, living a healthy, normal, healthy life to this day. So despite the remarkable success of Emily's story, CAR T cells have primarily benefited leukemias and lymphomas, and not the majority of cancer patients, especially those with solid tumors. At the time, I was working on engineering antibodies at a company called Genentech, and this story was very inspiring to me, both personally and scientifically, and I became convinced that CAR T cells were really the most viable strategy for achieving actual cures in cancer. So I went to the source of the story at the University of Pennsylvania and worked on engineering the next generation of CAR T cells. So to engineer a new CAR T cell, you need two main components. One is the knowledge of truly tumor-specific targets on the surface of a cancer cell, and the ability to engineer a receptor that can precisely recognize that target. So I want to draw an analogy between cells and houses to try to explain our work in targeting cancer cells. So at its core, a cancer cell is a cell that has gained the ability to divide uncontrollably. On the surface, because the cancer cell is derived from a healthy cell, it's largely indistinguishable from the healthy cell that spawns it. But if we look inside of the cell, the activities of a cancer cell are very different from a healthy cell. And that's similar to a house. If the inhabitants of a house are working on replicating their house, you might expect to find things like construction materials, plans for replicating the house, and specialized tools. And the similar analogy holds true in a cancer cell. However, the first generation of CAR T cells are only able to see the surface of the cell, so these activities are really invisible to these CAR T cells. And because of that, they target both the healthy cells that spawn the cancer in addition to the cancer cells. Now, in Emily's case, this was okay because she had a malignancy of B cells, and you can survive without your B cells. However, that principle doesn't hold true for solid tumors. In order to kill a brain tumor, you can't eliminate the brain. So the targets really need to be much more precise, and so that forced us to think very differently about how we approach solid tumor targets. So, as is well known to both archaeologists and intelligence officers, one way that you can really understand what's going on inside of a building is by examining its trash. And in the case of a replicating house, you can think of that as the construction materials, the blueprints for the house, and remnants of the construction process. And it turns out that the cell actually has a very analogous, remarkable system called HLA, where proteins at the end of their life cycle will be presented on the surface of the cell, basically presenting a snapshot of the internal activities on the surface of the cell. So we took advantage of this system to identify targets that were truly specific to cancer cells presented through this HLA system. And this is an example of one of the molecules we identified in neuroblastoma. And in the first column, this is showing the level of this molecule in cancer cells compared to healthy tissues. And on the left side, that's compared to a conventional cell membrane targeted by conventional CAR T cells called HER2. So through this process, we're able to identify targets with a much wider therapeutic window. And then once we find these targets, the second challenge was then to construct CAR T cells that can precisely recognize these cancer cells and not recognize the highly similar cells that they derive from. And an important analogy here is that we want to be able to differentiate a replicating cell from something like the house that's undergoing remodeling that might also have similar contents in the trash. And so through this process, we were able to identify unique signatures that are only found in cancer cells and not in the highly homologous cells that they spawn from. And this is what it looks like in practice. So these are CAR T cells cultured with on-target and non-target tumor cells. And in red, you can see the marker of tumor cell death. So the first of these is now in a clinical trial at the University of Pennsylvania and the Children's Hospital of Philadelphia. And we're looking forward to sharing the outcomes of this trial. So inspired by the therapeutic potential of this class of targets, we wanted to expand this approach to other cancer types. And when we started looking for these types of targets in other tumors, what we found is that there were both shared targets that we found across different cancer types, but also very unique targets that were particular to each class of cancer. And the analogy I want to draw is that for constructing different types of houses, you might find different types of materials in the trash. So brick houses, you'll find bricks, stone houses, wood houses, etc. And so we've been systematically mapping these contents of the trash across different types of cancer to identify targets across, so far, 21 different cancers. And so thanks to the support of Damon Runyon and the Bakewell Foundation, we've now been able to generate what we call Immunoverse, which is kind of an analog to the Google Maps of cancer targets, where we've identified 15,000 of these targets, and we're now pursuing therapies against a number of these different targets. So in summary, this type of therapy that we call peptide-centric CAR T cells allows us to target key drivers of cancer that are located inside the cell and expand our ability to target different types of cancer. We've identified a large number of these, both shared and private targets across 21 different cancer types. And that's enabled us and other groups to now develop new therapies in cancers that haven't previously benefited from CAR T cells. And we've made this atlas available to the scientific community to really try to catalyze the development of these types of therapies. And as another project that sponsored by Damon Runyon, we're now working on other ways to distinguish cancer cells from normal cells beyond the contents of the trash. And so, yeah, I just wanted to close by sharing my gratitude for the support. As you've heard, we're in a time of contracting support for the scientific endeavor. And I think as humans, at times of scarcity, the natural response is to retreat to safety. And so, we're really thankful to be able to do, rather than retreating, to do our most exciting work at this time. So thank you.
Thank you so much, Mark. So it is now, I'm absolutely delighted to introduce Dr. Craig Thompson, who has led a long and distinguished career in cancer research, including 12 years as the past president and chief executive officer of Memorial Sloan Kettering Cancer Center, where he still maintains a laboratory. Most importantly, he served on the Damon Runyon Fellowship Award Committee and also the Damon Runyon Board of Directors. He mentored 4 Damon Runyon scientists, including tonight's honoree, Dr. Matt Vander Heiden. He and Matt are both rare and unique physician scientists, trained in as both clinicians as well as laboratory researchers. Please join me in welcoming Dr. Craig Thompson to the stage.
Well, first of all, I made the mistake of writing out my notes, and there's no way to read them up here, so you're stuck with me. First of all, I suspect you're going to put me up here to be the bridge. So I am going to introduce Matt, but I also want to remind everybody in this room, because we're all connected to the Damon Runyon in some way, of the incredible success over 80 years that the Damon Runyon has had in bringing innovation into this horrible disease, cancer, to understand its underpinnings, to develop new therapeutic approaches and new diagnostic approaches to improve its diagnosis and treatment. But as Kelly just told us, to get ahead of the disease and actually deal with prevention or delay in onset of the disease. Throughout all of that, Damon Runyon has promised and delivered what they preached, which is innovation and new ideas, and sponsoring the leading edge. And the new ideas, as we've heard multiple times tonight, comes from young investigators. So we just heard about 2 people that when I joined the Damon Runyon's board would not have been possible to be funded with the funding that we had, that was started in 1946 to bring new scientific insights into cancer based on the original premise of the institution. It was the innovation that Damon Runyon's did that allows us to invest in engineering principles like what Mark just told us about, to bring new modalities, our own cells to bear on treating cancer. And what Kelly told us, innovative clinical trials based on the best in clinical insights to prevent cancer from ever taking hold in an individual. And that's really what emphasizes why I've been so proud to be part and why Matt's a really important recipient of this year's honoree in the dinner. And that is because in the first iteration in 1946, the Damon Runyon was dedicated to being real basic scientists into this problem of cancer. And I want to remind all of us, although we've all been touched by cancer or been involved in it directly, that in fact, cancer was a strange disease at the time. It affects every organ in our body. It's not like the diseases of the heart or the lung. It affects every organ of our body. And that made it very difficult for physicians to understand and come to grips when a patient came in, if they needed something different if they got prostate cancer than if they had lung cancer when they presented. If they had breast cancer, they needed a different diagnostic approach as it went through. And so the first iteration of innovation of the Damon Runyon was to actually convince and provide funding for new ideas from basic scientists, PhD trained, rigorous scientific principles into cancer. If that had not happened, we wouldn't have turned the tide on using toxic chemotherapy that began in 1990 and is playing out to today and has doubled our rate of being able to cure cancer and more recently tripled our rate of curing cancer as we go forward. And so what is interesting about the talks, the people that were chosen today, is that Bill and I represent a generation of physicians who are trained as physicians. We went to medical school. I get generously said to be a physician scientist, but I have no scientific training. I've never taken a graduate course in science. I took a medical school course in basic science. And it wasn't possible for physicians to get involved in basic research. So it's also ironic that the individual that convinced Bill and I to devote our careers to basic science principles of science is Dr. Livingston, David Livingston. David was a mentor for me about a decade before Bill was a fellow. I was an intern and resident for David. And David was the first person I ever met who actually understood science and could do a great job in rounds teaching us how to be physicians. And he told you could have a career like this. It took 9 years of retraining, so it's not an easy path. I never won any of these fellowships. And I can demonstrate why I can deliver on what I just preached, because I am a tremendous benefit of that generation of fellowships from the Damon Runyon. My wife, who's sitting over here, is an MD PhD, so she actually has the real credentials. She came to the United States from where she trained in the Karolinska to work as the first postdoctoral fellow for Mark Davis, who cloned the T-cell receptor. We wouldn't have CAR T-cells without the discovery of T-cell receptors and how they function. And she cloned the third chain of the T-cell receptor as it was done when she was a fellow, and she applied and got funding to do that work because she had a PhD. And so it was a way to salvage people that had decided to go to medical school. Before she got the funding, she was being trained to be a pediatrician. She's never looked back in the work that she's done in immunology. Fortunately, she did that work in the United States as an immigrant, as Bill told us about, and has delivered on that promise, and we've run a lab together now for the last 35 to 40 years. Okay, we're now going to turn to the second generation of innovation in the fellowship program, and that was to open it up to people that actually wanted to have a combined career as a physician and a scientist, and Matt is one of those representatives. And as you heard, he applied to a competition, and people that were on the board at the time, David, who's here and is being honored tonight, David Marshall, and Sam, and I were all on the board at that time, and we started to say, we need people that have had clinical training, have completed clinical training, who have taken care of cancer patients to actually be involved in the care of cancer patient and bring their innovations that combines insights from the clinic and insights in basic science. And Matt was an MD PhD student when I was at the University of Chicago with me and became interested in the idea of fully training as a physician, but at the same time having an opportunity to bring new principles to bear. And at that time, we were trying to understand why normal cells die off when they're not needed and cancer cells wouldn't do that. And that led us, without any stories, to trying to think about why cancer cells use the food in our body for different purposes, to grow and divide rather than to do any useful job. And that was the underpinning of this field now that Matt's a leader in, called cancer metabolism. The most difficult thing I had to do in Matt's training was to convince him at the end of his PhD not to drop out of the MD PhD program. And that was because he was convinced that all of the insights that we were gaining from cancer metabolism would be done by the time he went back and finished his clinical last 2 years of medical school and did his residency, and it would all be over, and he would have missed the single most important thing he had ever done. And I just said, no, it's not going to be. If you and I are the only two people talking about this in the world, which we might have been at the time, it's not an important problem. And if it is an important problem, we're not going to solve it all in just a brief period of time. And fortunately, we were able to convince him to go on to train at Harvard, and first internal medicine, and then at the Dana-Farber as a medical oncologist. And then he came back and said, how am I going to get this funded? There's no way to fund a clinical fellow to do basic science research. And at that time, the people at the Dana-Farber were able to say, no, there's this Damon Runyon program. David Livingston's been talking about it for years. It's all about getting people back into the lab with their clinical insights. And so Matt joined a laboratory, Lou Cantley's lab, restarted up his interest. Lou had also come to the conclusion that the oncogenes he was studying in the PI3 kinase pathway were in fact involved in cancer by a metabolic approach. And so he was able to pioneer some of those early studies in Lou Cantley's lab. Why this is innovation that spurs to what the two talks you just heard is that Matt, at the end of his fellowship, decided that it was time to actually, the difficult thing about people in metabolism was that people couldn't see the connection. And at the time, the first gene that was a metabolic gene that caused cancer, a mutation in a metabolic gene that caused cancer, was the gene Kelly just told you about. It had just been discovered in brain cancer in young people, that the mutations in this IDH gene, which is the isocitrate dehydrogenase gene, for those that have had a science course in the room, that's a gene in the Krebs cycle that regulates how we burn stuff to make ATP. And when it's mutated, it creates an abnormality that caused cancer. And so Matt did a gap year. Before he went on to be a faculty member at MIT, he decided to join a team of people to investigate how it was that a mutation in a metabolic gene could cause cancer, and discovered the first oncometabolite. With that insight, because no one believed it even when it was published in Nature, he went on to start his own independent laboratory at MIT. He teaches the core course for undergraduates in biochemistry and has revitalized that field going forward because he's a tremendous mentor and a tremendous teacher. That's the wisdom that David Livingston convinced us about. But more importantly, he went on with that group of people and to discover a drug that Kelly just told you about that works in leukemia, but much more importantly was able ultimately during the pandemic to be involved in that drug being evolved into a drug that crossed the blood-brain barrier to treat kids, actually not kids, 20-year-old and 30-year-olds that had the most common form of brain cancer that exists in young adults. Everyone in this room knows somebody in high school or college that had an intermediate grade, what's called an intermediate grade glioma. That's much like what Kelly said. You get an abnormality of your blood counts when this happens in your blood cell. It's what happens. This gene is a single gene, just causes an abnormality in growth in your brain cells. And a clinical trial that was said not to be able to be done was done by innovative clinical scientists, of which Kelly was a member of the team broadly at Memorial Sloan Kettering at the time, completed a trial worldwide that demonstrated that it delays by at least 7 to 10 years the onset of the disease, sadly, that Bill Kalin told us about with his wife, glioblastoma. Because why those young people go on to die of cancer is because they transform into glioblastoma. That drug was approved last September in the United States. I actually decided about a month ago that we should find out whether actually young people that have this diagnosis, they know they don't have cancer, they're being watchfully waited by their neurologist, waiting for it to progress so that they'll get the treatments which are not good in glioblastoma. There are about 20,000 like that. In the first 6 months that the drug was available in the clinic, there are 5,500 young adults on that drug right now being given hope. The original trial cohort, more than half of them in the last 6 years have gone on to have glioblastoma. And they have not yet even come close in the treatment group. So we will wait and see. But that's the kind of hope investing in physician scientists. And then the innovations that Kelly's told you in other diseases around this that are going on. So it's an incredibly exciting time in science, despite what Bill reminded us about in terms of some of the issues and some of the challenges of convincing people that science delivers for the benefit of our health. But it's a real privilege then to turn to really what we're here for at the end of this ceremony, which is to honor Matt Vander Heiden. Today, he's not a young investigator anymore. He's not funded anymore by the Damon Runyon. He gives back by being on review panels. But what's really important is because of his training, he is one of the leaders. He's one of the best mentors, as Bill told us about, in our field today. As one of the investigators I won't mention that's at our table reminded me, he's the mentor of mentors in our field. He makes everybody better. He gives us the best advice. He is the glue that makes these new innovations in metabolism work. And for that, he deserves to be honored. But beyond that, he's also the director of the most important basic science cancer center funded by the National Cancer Institute, the Koch Institute at MIT, where he really spurs innovation that brings engineers, that brings other investigators from other fields into cancer, which is what MIT is so well known for. And Matt is now the leader, having taken over that work from others that have championed that. And then finally, because he's a triple threat, he's a leader in maintaining our voice as America continues to lead the world in biomedical innovation. For that work, he's been elected to the National Academy of Medicine. He was recently elected a fellow of the American Association of Cancer Research, AACR, where his voice is really important and reminding us that it's young people that make innovations and that our ability to continue to support and mentor them is what will keep America a leader in world health. And so with that, it's my privilege to introduce Matt Vander Heiden. I couldn't be prouder of him and all of his accomplishments. Matt.
Thanks so much, Craig. So we have, Damon Runyon has a program now for MD onlys to be able to go. It's a committee I serve on. We would have taken you if you applied a long time ago. It's always hard to follow Craig, I'll tell you that. All right, I just want to start off by saying how humbled and honored I am to be recognized tonight, especially by such an important milestone celebration here at the Damon Runyon Foundation, an organization that I deeply, deeply respect and have played a really formative role in my career. Damon Runyon invested in me at a time, as you heard from Craig, when I was far, far, far less accomplished. So I came from a relatively small town in Wisconsin. I was always interested in science. I knew growing up I wanted to make an impact, but like many young people, I had no idea how to do that. I know that I needed to work hard. I could do that. I did that. I still do that. But we all need mentors to guide us. And I can't tell you how fortunate I am to have found Craig and worked with Craig. He was actually much less of a big deal then, although he was the same person, I promise you. But Craig, I'm really grateful to you for teaching me how to think about science, how to think about leveraging discoveries to make impact, and also for helping me make some good decisions, like finishing my clinical training and going on to becoming a medical oncologist. I also want to thank you for helping me position myself to work in a nascent field, this intersection between cancer and metabolism. As Craig alluded to, at the time, it was something not a lot of people were doing, and I would dare say interested in. I still remember as a PhD student giving some of my talks on it, people sleeping. Hopefully no one's doing that now. The lights are bright enough I can't see. But then after Craig's lab, I did, with Craig's advice, I finished my clinical training, as you heard from him. I did a postdoc. I had lots of ideas. But I also had to confront the realities. I was a junior oncologist. I was in a hospital system. And like all businesses, hospitals have to pay their bills. From a business and healthcare perspective, at that time, I was far more valuable seeing patients than I was. That paid my salary. Chasing some crazy ideas that were certainly not mainstream in metabolism and cancer at the time was, maybe not from the business standpoint, the right decision to make by my division clinical leaders. So to keep doing science, I needed some funding. And so what do you do when you need funding and you're a scientist? Well, you write a bunch of grants. And I wrote grants and I applied to Damon Runyon. And I still remember the day they called. I was sitting at my bench. This was still, we had phones in the lab and whatnot. And I answered the phone and I was told by the program officer at Damon Runyon at the time, Young did this job immediately after the person who called me. And she told me I got a postdoc fellowship and she asked if I would accept it. At the time I was like, yeah, of course. It was really the first validation that maybe I could support myself in a research career. That was cool. And Lord knows I needed the funding, okay? What I did not realize then, but would soon learn, was how transformative being a Damon Runyon Fellow really would be. So beyond the funding, and believe me, that was critical, especially at that time, but they also provided a community of really amazing fellows. Now, many of these people did impactful things. Some of them are actually here tonight. And I have to say, some of you could be up here doing this as well because you have done some really amazing things as well. They also provided access to some really incredible mentors, incredible scientists, people like Bill Kalin. And Bill, you've been a great source of mentorship and support for me over the years too, and I'm very, very grateful to that. Now, tragically, this experience actually allowed me to experience firsthand exactly what it is we have to do. The program officer at the time, the person who preceded Young, unfortunately, she has a story very similar to the one that you heard from Young and that you heard from Bill. Her husband, way too young, died of, I think it was osteosarcoma at the time. And, when that happened, it really came home that, the work we do for all the successes, right? In science, you get wrapped up in the successes and everything else, but those are reminders that the patients need us. Patients need better solutions, and it is our responsibility as scientists. And I remind my lab about this all the time, and I also remind my colleagues at MIT and the Koch Institute all the time as well, that what we do matters because patients count on us, and you guys allow us to be able to do that. Now, beyond the validation and support of being a Damon Runyon Fellow, was it also helped me to ultimately, when I started my own lab, start to develop my own scientific portfolio. And this actually led to a call from Damon Runyon again, where I got the Rachleff Innovation Award. And I want to thank Andy Rachleff for supporting that program because that also made a big difference. But I have to say, through all of this, maybe the most important call I got was actually one I got from Phil Sharp at MIT when he called up and said, hey, we want to hire you at the Koch Institute. And that allowed me to build a thriving lab. It allowed me ultimately to become, what you heard from Craig, Cancer Center Director about 5 years ago. Now, all this support was critical. It allowed me to establish my young lab. And as Craig alluded to, these investments paid off. Now, I'm really proud of the work and the contributions I did. Craig was very kind to list some of them. I feel very lucky and special to be involved in any way in development of drugs that actually helped real people. Not many people in our field can say that, so I feel very lucky to have gotten to do that. But I have to say, the impacts that at least I think I've made really didn't come because we set off, oh, we're going to make this drug. They actually came because we were doing basic discovery science. I was asking questions. I wanted to understand how metabolism worked, but it was really my physician scientist training when we found something new. It was like, wait a second, it works this way. Maybe that's a connection that we can now leverage to do something different for patients. And it's really those types of insights, in my view, that impact cancer the most. Now, Damon Runyon's been very important to me over my career. They've had tremendous impact, and for that reason, I've always been really happy and opportunities to give back. You heard from people. I've served on several award selection committees. I continue to do this, not because, like Bill, I don't have infinite time. He has less time probably than I do. But this is an organization that gets it right. And if I'm going to spend my time on things, I want to spend my time on things that actually matter. And Damon Runyon is one of those things. And that's because they recognize that we need to invest in basic discovery research, even if it's less fashionable in recent years. So this is more important forever. And that's because we need to fund discovery because, well, translation is obviously important. And look, I believe in translation as well, but new solutions require new knowledge. Innovation of tomorrow depends on discoveries that are made today, right? We can translate what has been discovered in the past, but if we're going to have the discoveries and the translation of 10 years from now, we need new discoveries today to fuel those future innovations. Damon Runyon also recognizes that they need to invest in talented young people and do so at key points in their career. I agree with Craig. I am no longer a young person. I would never claim to be. But I learned through my own training, and I try to pay this forward, is really the importance of mentorship. And Damon Runyon cares about that too. They really want to steward the investments that they're making because they know that that also matters. And they recognize that building stellar scientific communities has tremendous impact. If anyone out there ever wants to be inspired or humble, just, Young invites you to the retreat, go to the retreat. It's quite an experience. Now, Damon Runyon is honoring me tonight, and those who know me well know I'm not one who likes the spotlight necessarily. But especially I want to say something, and that is, while I'm maybe being honored, science is a team sport. My success is, of course, not just my success, it's the success of all the people who have collaborated with me, all the trainees in my lab, and really this honor and any honors we get as scientists often reflect the work that's done by our trainees. I've been very lucky to work with many, many amazing people over my career. You heard some of them have been supported by Damon Runyon. They make good investments there too. And I suspect at the end of the day, my biggest impact in science is really going to be the people I trained and I helped mentor. And it's, Damon Runyon gets that, and I think that's really special. But maybe what's most remarkable about Damon Runyon is, whatever you think of my story, it's not at all unique. There are literally many of you out there that I respect deeply, and I think you could be up here giving this exact same talk. Damon Runyon has fostered the careers of so many exceptional scientists. I don't think we could even easily quantify it. They probably have, but I don't think you could be. They have this really uncanny ability to spot and cultivate talent, and that impact is just incredible. Now, unlike many organizations, I think one of their secrets is that organizations like to fund projects or specific things. Damon Runyon, at the end of the day, they fund people, right? They fund people with good ideas, and those people have projects. But at the end of the day, they make bets on people. And for anyone out there who's ever been involved in a business or running an organization, people make the difference. And this is true in all fields, and I think the success of their strategy really shows. Now I want to close, and I just want to close by thanking Damon Runyon again for all the support of science, all of you who have supported Damon Runyon. I'm really marvel at the legacy that they've created over the last 80 years. But specifically, I want to call out all the people that work at the foundation. As I said before, in any organization, it's people who really make the difference and drive success. And again, we all know organizations can sometimes lose their way. Sometimes success can lead to bureaucracy. It can lead to people making decisions where the things that led to their success are not the things that are focused on. And because of the people at Damon Runyon, who I love working with all of you, Young said that, I honestly view you guys all as friends. I really value our friendship. But you guys have somehow found a way to never lose sight of what's important, to always sort of stick to the course, and that is do what's right to foster good science. You foster great science, invest in good people, that is going to make a tremendous difference. So for everyone who's affiliated with Damon Runyon, everyone who's donated to Damon Runyon, thank you very much. Thank you for honoring me, and congratulations on 80 years.
Thank you so much, Matt. Thank you again to Drs. Kalin, Bolton, Markovich, Thompson, and Vander Heiden, our extraordinary speakers this evening. Thank you to our board of directors and our amazing Damon Runyon team, especially Kim Kubert, wherever she may be, who puts together all of the details for this event. And to all of our guests for joining and spending this evening with us. We're immensely grateful to you for your support and dedication to our important mission to continue the Damon Runyon legacy of funding brave and bold. In this pivotal moment for science at large, we in this room have an opportunity to ensure that the best and brightest stay in cancer research. We need to do all that we can to enable them to remain focused on their important work and to improve treatment, diagnosis, and prevention of all cancers. We owe it to these brilliant scientists who have dedicated their lives to bettering the world, to our loved ones, and to the memories of all those who have lost their lives to cancer. Your generosity allows us to increase our support and impact at this critical time. This year alone, Damon Runyon committed nearly $22 million to brilliant young scientists, the future leaders of their fields. So thank you for enabling us to support the next generation of brave and bold scientists. So, this concludes our program, and we invite you to stay for a dessert reception, which is set up outside, and mingle and meet more of our Damon Runyon scientists. As you leave the event, please pick up a special gift that's been provided by our board of directors to commemorate our anniversary. Thank you again for celebrating with us, and please enjoy the rest of your evening.